Welcome everyone. I'll start today about a breakthrough type of medication, which is the monoclonal antibody. How is it made and how is it used? So, monoclonal antibodies, basically, the idea here is by combining a beta lymphocyte with a tumor cell. As we all know, beta lymphocytes are the type of white blood cells that can make antibodies. Tumor cell has the ability to divide rapidly. So the idea is by mixing the beta lymphocyte with a tumor cell, I'll have a cell that's able to divide rapidly and in the same time produces antibodies. This cell is called hybridoma, because it's a hybrid of the beta lymphocytes along with the tumor cell. So basically, what we get here how we do the technique is we inject the mouse with the chosen antigen, the antigen of interest. Now the white blood cells of this mouse, the beta lymphocytes, will start to, or it is ready already to make up the antibodies against this particular antigen. All right, and then we get fast dividing tumor cells. We create them in the lab. We get the tumor cell and fuse it with the beta lymphocytes to make the hybridoma. Now, hybridoma can be cloned to get a lot of identical cells. These cells all produce the same antibodies, the monoclonal antibodies. The antibodies can be collected then and purified. This gives the antibodies quickly, and also it's a cheap way to, not very cheap, I would say relatively a cheap way to produce it. So you can make monoclonal antibodies that bind to anything you want, an antigen that's only found on the surface of one type of cell. Now, the thing with monoclonal antibodies, it will only bind to the target molecule, which is the antigen. So it will be specific for, I would be specifically binding onto the antigen or, the antigen of interest. Now, what uses of monoclonal antibody, or how could it be used? Actually, it has wide uses. One example here they choose for the EQAGCSE is how to use monoclonal antibodies as to test pregnancy or as a pregnancy test. So basically, maybe we have seen before the pregnancy test. It's a strip, okay? It's a strip where you pee here in this square, and then the test result shows on the other square there, okay? So the idea, when you pee on this window contains an antibodies that's stuck to a blue color or a blue bead. Okay? Then if you're pregnant, there is a hormone called human chorionic gonadotropin, or HCG, human chorionic gonadotropin. Okay? These antibodies on the blue beads are made to stick to the human chorionic antibody. It's made to stick to it. Okay, so as you see here, that's the human chorionic gonadotropin got bind to the antibodies. And then as you pee, the fluid will carry the antibodies along with the HCG hormone. Okay, you don't need really to know that the long name, the human chorionic gonadotropin, HCG, is enough. So P will move on until it reaches to the test strip, okay? So the test strip contains antibodies that are immobilized, unmovable, okay? So these are antibodies stuck down here on the test strip. That's the test strip. So these antibodies will immobilize the blue beads only if there's HCG available, only because it will bind to the immobilized antibody. With these blue beads, this will immobilize the blue beads here on the test strip region. So we will be able to see a blue color, okay? So seeing this blue color means this person is pregnant, because the presence of the HCG is what allows the blue bead to be stuck on the immobilized antibodies on the test strip. Otherwise it won't. So if we check the other case in here, if the female isn't pregnant and then HCG isn't present, then the beads carried within the flow, as I said, to the test strip area, see, it won't get stuck here onto the immobilized antibodies, see? So it will just move away and be washed away. It won't stick. As a result, the test will show no blue color. The test will show no blue color. That means this female isn't pregnant. So that's one of the applications of monoclonal antibodies. Moving on to another application. So monoclonal antibodies can be used to treat diseases as well. Okay, so we know that different cells in the body have different antigens on their surface. So if I'm talking about a normal body cells, that these are the self-normal antigens on the cells. Cancer cells are different cells. Therefore, the antigens present on the cancer cells are different than the antigens present on the normal body cells. These antigens present on the normal cells are called tumor marker. The one present at the cancer cells are called tumor marker. So if we're able to design a monoclonal antibody that's specific and complementary to the tumor marker, so these monoclonal antibodies must bind specifically to the tumor marker and hence the cancer cells. What if we're able as well to place the anti-cancer drug attached on these antibodies? Then these antibodies are now used to deliver this medication specifically onto the cancer cell, and so can damage the cancer cells without affecting, or killing the cancer cells without affecting any of the normal body cells. So it's great to use the monoclonal antibodies here as a target therapy. Target therapy, because it targets a specific antigen. I hope this part is clear. That's a very important application for monoclonal antibodies. So depending that it's targeting a specific cell. There is as well many other uses for monoclonal antibodies. So monoclonal antibodies can be used to bind to hormones and other chemicals in the blood to measure their level. They can test blood samples in laboratories for certain pathogen, as I said, if they're made complementary to this pathogen. They can locate the specific molecules on a cell or in a tissue. So they're used as well, guys, for diagnosis. Okay, not only treatment, also for diagnosis. How is that? So the monoclonal antibodies are made that they will bind to a specific molecule that you're looking for. Then the antibodies are then bounded to a fluorescent dye, something that would show a color, like fluorescent color. If the molecule of interest are present in the sample you're analyzing, then the monoclonal antibody will attach to them, and then they can be detected using this fluorescence. So if you see a fluorescence, that means this molecule of interest is present, okay? And if not, that means it isn't there. So this is a good use for diagnosis. Now, monoclonal antibodies, although the important or the lot of positive applications it has, it also comes with problems or some cons, right? So there are some obvious problems from using monoclonal antibodies. One big one is the cancer treatment. Other cancer treatments like standard chemotherapy or radiotherapy can affect the normal body cells as well as killing the cancer cells. But monoclonal antibodies only target specific cell, right? So that's a big pro. However, there are also side effects. Okay, so the side effects of the monoclonal antibodies, well, actually, they have some more side effects than what was expected. So they cause fever, they may cause vomiting, low blood pressure. All right, so that's also other side effects it may cause. This means that they are not as widely used as treatment as the scientists originally thought. Okay, because of the side effects that it may develop. I hope that was a beneficial lesson, a quick review for monoclonal antibodies, how's it made, how's it work, and also the pros and cons from using monoclonal antibodies.